Okay. There was a request for a sort of sum everything up before the test, please video. So we'll try to do this like chemistry and DNA 101 in less than 30 seconds. So we're going to go through the macromolecules and then I'll swap over to the genetics slides and we'll run through those super fast. So I'm just scrolling down here. Okay, macromolecules. All right. So the macromolecule slides are actually on the end of the Unit 2 chemistry notes. So you'd have to go back in Google Classroom and go to the unit notes and then go Unit 2. And it starts on slide 64. So it's 64 for me, but if you have your own video notes or your own electronic notebook, then it might have gotten pushed back as you added things. So macromolecules, they're large molecules, meaning they are way bigger than anything else that we've talked about in class so far. So things like methane and hydrogen gas and water would all be really, really small. And the macromolecules are really, really big, big enough that we could see them without a microscope, some of them anyway. So there are 4 macromolecules. They're also called polymers. They're also called biomolecules. They're also called organic molecules. And if you want to review any of those, the Edpuzzles are still up, so you can go back and kind of rewatch those videos. There are 4 of them: lipids, carbs, proteins, nucleic acids. Lipids were the fats, waxes, and oils. They make our cell layers, cell membranes. They're non-water soluble. They're E-shaped. We also talked about ones that have less than 3 tails, so only 2 tails. We talked about their monomer parts, which for lipids are not true monomers. But if you were thinking about a monomer part, there would be the glycerol in the circle here. And then these trails are called the fatty acid chains. So non-water soluble, used for insulation, protective layers, things like butter, oil. We did the Vaseline on our hands. We did the fat mittens. You guys have mixed water with oil and then tried detergent to see how that kind of breaks down the oil molecules. So that's what they are used for. Carbs are our quick energy, water soluble, so we chewed up the crackers. So you'd see that they were dissolving in the water, and then when you try to eat them, they dissolve super fast in your mouth, and that's because your mouth has enzymes, which are not carbs. Those are actually proteins, but it was just to show that they kind of break down very easily for you. So this would be a polysaccharide chain or a carbohydrate chain or a starch. And when we usually see the monomers, it would look like this. And this is one of our carbon ring shapes. This particular one would be glucose. And our formula for these are always in a 1:2 to 1 ratio. So we wrote down glucose C6H12O6 and talked about how if there's 1 carbon for 2 hydrogens for 1 oxygen, that very special ratio means that you're looking at a sugar. For us, these are energy sources, but for plants, they're usually structural, making up the cell walls. Plant sugar is glucose. Our sugar is different than glucose. It would be glycogen. Plants store it in starch, and we usually break down the starches to make our own energy when we eat things like bread and pasta and potatoes, root vegetables, sugars in fruits and juices and things like that. Last one we talked about before we got into DNA was proteins. So proteins are usually structural. There are 2 different kinds of proteins that we generally think about. One is, in fact, the structured ones. That would be our hair, our nails, our muscles, our bone, our cell shape, our pigmentations, and some of our hormones. And then the other version of proteins that we see are the enzymes, which are catalysts. So the reason you were capable of chewing up that cracker in less than a minute and not having very much cracker left is because there's actually an enzyme in your spit called amylase, that was breaking down that cracker for you. And then we talked about the fact that they are blob shaped like this one, but that they have to be very special blobs. And if they're not folded in just the right shape and bonded in just the right shape, they don't work. So we kind of used our little origami things to demonstrate that for us. Within this blob structure, there would be little tiny pieces of these proteins, the monomers of the proteins, which would be called amino acids. And there are actually only 20 different kinds of amino acids. And when you arrange those 20 different kinds of amino acids in these very long chains, they end up making all sorts of really diversified protein structures. The last of our 4 macromolecules were the nucleic acids. There are only 2 different kinds of nucleic acids. One is DNA, one is RNA. For all the other different macromolecules, there are multiple different forms of the molecule. So for example, for the carbs, we have sugars, we have starches, we have fruit, we have pasta, bread, root vegetables, etc. For fats, we have oils, tar, lipids, wax, earwax. That was the answer to that question, by the way. Um, vegetable oil, animal fat, Crisco. I mean, you could keep listing them. And then for proteins, there's the skin, hair, nails, bone, muscle, enzymes, hormones, everything else. And then we come to nucleic acids, and there's only 2. There's DNA and RNA. But we've already seen that the DNA with only its one form is so advanced and so intricate that we are actually going to spend a whole nother month just talking about DNA and what traits are and how the traits are coded for and how your cell goes about organizing its DNA and all of those awesome things. So here's the DNA molecule. You guys, I think by now, and I hope by now, feel confident with the fact that the sugar and the phosphates are on the outsides and the nucleotides have the nitrogenous bases on the inside. A only bonds to T, C only bonds to G, phosphate's the circle here, the sugar is our little house right here. If you have one phosphate, one sugar, and one nitrogenous base, that's called our nucleotide. The A's, T's, C's, and G's going across the center are the rungs of the double helix ladder, and the sugar-phosphate backbone, or the upright sides, are the outside of the double helix, and our code is encoded in the order and the length of the G's, T's, C's, and A's. We talked about Rosalind Franklin. We talked about Chargaff. We talked about Watson and Crick, who were the 2 guys who stole Franklin's data. And then we talked about how DNA is wrapped up inside the cell. So if you look at one cell and you have the loose chromatin, which is as the DNA usually exists in the center of the cell, and you pulled that out, it would actually be 6+ feet long, so taller than you. And that when your body is ready to move it around, it will take the DNA, so here's the loose DNA right here, here's a base pair, and it will wrap it around proteins called histones, and then it will pile those histones up, and that will become a chromosome. This particular picture has 2 chromosomes joined together, so here's one on the right, here's one on the left. This is still called a chromosome, so 1/2 of this is called a chromosome. When there's 2 duplicated, that is also called a chromosome. That duplication portion is not on your test. That got pushed to the next test. The center is called the centromere. And if you're looking at a duplicated chromosome, 1/2 would be called the chromatid. Again, the chromatid and the fact that this X is the duplicated chromosome is not on this test. We pushed that forward to the Unit 4 test. So we will come back in February and start talking about this. So what you need to know from this picture, we drew in class on the board. The picture is still up on Google Classroom for you, and it's basically the idea that the DNA wraps itself around the histones and that a segment of DNA is what's called a trait, and that the traits tell our cells how to make the proteins that then take on our physical form and make our skin, our hair, our nails, so on and so forth. So there's the last month in 9 minutes and 20 seconds, and I hope that helps the person who requested this. Thanks, guys.